We'll continue our notes on biotechnology by discussing problems with genetically modified organisms and also talking about gene therapy. So GMOs, or genetically modified organisms, have one or more genes that have been introduced by artificial means. Whether the gene is from another organism, whether it's one that has been altered in some way, that means it's genetically modified. Transgenic organisms are ones that contain at least one gene from another species, but there are other ways that genes can be modified besides from another species. Nowadays, we're producing plants, for instance, that are either more resistant to herbicides or pests, or help provide additional nutrients that might help address malnutrition in some areas. There are also GM animals that are being produced that, for instance, can produce milk that contains antibiotics or vaccines for certain kinds of diseases, additional nutrients of various kinds. We have to watch this very carefully. Scientists use a great deal of safety measures to guard against this, but there are still concerns in the public about GM organisms, including the potential to accidentally introduce allergens into the food supply, or perhaps accidentally have genes spread to a closely related species. There are regulatory agencies that work on this all the time, trying to address the safety of these products and labeling them properly and make sure that the biotechnology is being used safely. Another thing that DNA technology is used for is gene therapy. Gene therapy is an attempt to treat a disease by providing a functional allele for one that doesn't function properly. So one way that you might do this is maybe to get a clone of a functional allele, inserted into a retrovirus. Remember, retroviruses have the ability to take RNA and change it into DNA. So you would use an RNA copy of the gene, put it into a retrovirus, use the virus to infect the cell type that you're looking at, for instance, bone marrow that's grown in culture. It would infect those cells and insert the DNA, the new DNA, the new gene into those cells. And once the cells grow with the infected part that include and reproduce with the added gene to them, then you could return the cells to the patient where they would grow and divide. And hopefully they would help cure the condition. It's basically altering the inflicted individual's genes and try to treat the disease. It works best when you're trying to treat disorders traceable to one single defective gene. For instance, you can see lots of things that are just one little change to one thing that can make a major problem. For instance, sickle cell anemia is one thing that is caused just by one change in one codon in the gene for hemoglobin. And so if you could engineer a corrected one and get it into the patient, then perhaps they could make appropriate kinds of cells and be cured of the disease. I don't think that one's possible yet, but that's something I think they're looking at. So here's kind of how it works in pictorial form. Okay, here's our cloned gene, our RNA version of the gene. We're going to insert it into a retrovirus, okay, which includes reverse transcriptase. The viral DNA would be infected into the cells and would insert into the cell's chromosome, grow those cells in culture, and then return the engineered cells to the patient and hopefully cure the condition. The first successful human gene therapy trial was in the year 2000, not that very long ago. They were trying to treat children that had a condition called severe combined immune deficiency. These are children that basically do not have much of an immune system. So the treatment that they do is similar to what we just went through, helped 9 of the patients, but caused leukemia in 3 of them, and 1 child died as a result of the therapy. And so there are things that, it sounds like it's pretty good if you have a 90% success rate, unless your child is the one in 10 that might not be helped by it. So we have to think about a lot of things before things like this are attempted. For instance, how can we build in control mechanisms that make sure that the gene is going to make the right amount of the protein at the right time? How can we do this without harming other parts of the cell or other functions of the body? And a big concern for a lot of people is, will this lead to efforts to control the genetic makeup of human populations? Because that definitely could have a drastic effect on the future of our species if we can physically and electively choose to change things like that. Organismal cloning is cloning whole multicellular organisms. This is often used in the production of stem cells, okay, because under the right kind of circumstances, stem cells can become any kind of cell. And so this has great potential for growing new organs for transplantation or things like that, specific to the patient. Now, it's easy to clone plants. Plants, if you grow a plant from a cutting from a different plant, that cutting, the new plant, is going to be a clone of the original plant. But the first single-cell cultures of plants was actually done in the 1950s at Cornell in New York. It's pretty easy to grow whole plants from differentiated plant cells because basically when you grow them in culture, they de-differentiate. They go back to more of like a stem cell. And so this is used extensively in agriculture, as you might suspect. There's some plants that reproduce best by cloning, for instance, orchids, also seedless grapes and things like that. It also lets you mass produce plants that have special characteristics. And so this is used quite a bit in agriculture already. Now, cloning animals is a little bit different because animal cells, differentiated animal cells, don't really divide well in culture, nor do they produce multiple cell types. If you can get them to grow in culture, they'll produce the same kind of cell that they are, but not too many other things. And so what scientists figured out pretty early on was that nuclear transfer might be the answer for that. So if you transfer a differentiated nucleus into an egg cell from which the nucleus has been removed, we call that enucleated, then perhaps that egg cell, which is programmed to develop into a whole organism, might work. The first experiments on this kind of thing were done in frogs back in the 1950s and 60s. They found that if they transferred a nucleus from an early embryo, let's say the 4 or 8 cell stage, you had a very high percentage of success, but the older the embryo was, the later it was in the development process, it was much less successful. Here we have what they found about frogs. Okay, less differentiated cells, most often developed into tadpoles. But if they took a fully differentiated cell, for instance, most of the time the embryo would start forming, but it would stop developing before the tadpole stage. And so they were less successful with more differentiated cells. Other things showed that there was some more success with embryonic nuclei, but not with fully differentiated ones. And so there were a lot of work that had to be done on this. Finally, in 1997, the first mammal was cloned from a differentiated nucleus. That happened in 1997 in Scotland by a scientist named Ian Wilmot. And basically what they found was if they cultured the differentiated cells into in a medium that was poor or low in nutrients, the cells became less differentiated. And so then if they took a nucleus from that culture and put it with the enucleated egg, they could try transplanting the embryo to a surrogate. And lo and behold, they ended up finally with a sheep they called Dolly. Here's how it worked. There were 3 different animals involved. There was the cell donor. Here's the mammary cell donor. The egg donor, a different sheep, and then the surrogate mother in which they implanted the embryo. So they took cells from the mammary gland of the donor here, grew them in culture in that nutrient-poor culture medium. They took an egg cell from the egg cell donor and removed the nucleus from it, fused these 2 cells together, and then they grew that in culture to make an embryo. Once an early embryo was produced, then they implanted that in the uterus of a 3rd sheep, and then you had a lamb, Dolly, that was born, whose DNA profile was identical to the mammary cell donor. Now, the reason they use 3 different sheep, of course, that's part of the control of their experiment. Remember, when you do experiments, you have to have controls, and so using the 3 different sources of the genetic material and the egg cells and so forth was part of the control mechanisms of the experiment. Now, what happens when you try to clone animals is that most trials actually don't work. Okay, there are more failures than successes. And a lot of the cloned animals exhibit some kind of defect, either metabolic disorders. Sometimes they develop age-related problems at a much earlier age. That's what happened with Dolly. She actually died relatively young for a sheep. The differences that occur there are probably due to epigenetic changes. Remember, there are changes that occur to the DNA in the lifetime of an organism that involve adding various chemical groups like methyl groups and so forth that can affect the pattern of gene expression. And so that's probably what part of the problem is. But I think they're still working on that. It's probably become better at it. There are a number of different animals that have been cloned, like cats and dogs and cows and various things like that. So they're more successful at it than they used to be, but there still can be some problems along the way. Now, most cloning of human cells is directed toward producing stem cells. Stem cells are undifferentiated cells that can grow into many different types of cells. Okay, these are called pluripotent cells. Embryonic cells are the ones that are pluripotent. Adult stem cells are less pluripotent because they're more specific to a certain kind of tissue. But with the right culture conditions, adult stem cells have been made to differentiate into different kinds of cells. So this is also something that's highly controversial, especially the use of embryos for production of stem cells. And so there are a lot of controversies that are dealt with in the production here. Embryonic stem cells, when you grow them in different conditions, adding the right kind of chemicals at the right time and so forth, can produce very different kinds of cells. Because after all, an embryo can make every different kind of cell there is in the organism. Whereas adult stem cells are usually certain kinds of cells can be made. For instance, bone marrow stem cells can make various kinds of blood cells, but not a whole lot else.